Good morning, everyone. Myself, Dr. Kundan Kumar Chaubey, Head of School of Basic and Applied Sciences, Sanskriti University, Mathura. I am going to discuss about the real-time polymerase chain reaction, is also known as quantitative PCR. So first, we know about the qPCR workflow. So for qPCR workflow, first we have to synthesize the primer for the real-time PCR. Then we have to send it to synthesis and procurement to the synthesizing company. Then after that, we are procuring the PCR reagents, PCR plastics, and then we set up the reactions. We set up the reactions inside the real-time PCR instruments, and then after when we getting the result, we analyzing the result using the different PCR analysis software. If sample is positive or negative, the genes are upregulating or downregulating, like that. So first of all, we know about the real-time PCR. So real-time PCR is a technique used to monitor the PCR amplification in real-time manner. Means we can observe, we can monitor each and every cycles of the PCR using the real-time PCR. At the same time, relatively we can know about a small amount of the PCR product, and we can also quantify it in real-time manner. So because it is based on detection of fluorescence produced by a reporter during the amplification, so there is no need of other accessories like gel running and gel documentation. So these types of things not needed in real-time PCR, which are required for the normal PCR. So now we know about the different PCR terminology we are using. So first is polymerase chain reaction, that we abbreviated it as a PCR, PCR. Second is reverse transcription polymerase chain reaction. We abbreviated it as a RT-PCR. Third is a real-time polymerase chain reaction. We abbreviated it as a qPCR. And fourth one is a RT-PCR, qPCR combined techniques. We abbreviated it is a QRT-PCR. Actually, RT-PCR is a two-step process. First is reverse transcription, and second is the PCR. In reverse transcription, we making the cDNA from mRNA, and in PCR, we synthesizing the double-stranded cDNA from the single-stranded cDNA. That's why we calling it as a RT-PCR. Then we know about the different steps of polymerase chain reaction. So first step is denaturation. So we are using the 95 to 96 degrees Celsius of the temperature for the denaturation, melting out of the both of the strands of the DNA in single strand. Then we go for annealing at normally at 68 degrees Celsius, but it depends on the GC content of the primers. And so to optimize the annealing temperature of the PCR, we are going for the gradient PCR. And third one is the elongation. So we are generally using the 72 degrees Celsius temperature for the elongation and extension of the PCR. So now we know about the fluorescence detection system using in real-time PCR. So first we have to manage all the ingredients required for the real-time PCR. Then we have to mix the PCR master mix. Then we have to put the samples inside the machine for amplification. And during amplification, we exposing the plates, we exposing the reaction by the UV light. And if amplification is going on, then after exposing from the UV light, it releasing the green light fluorescence that is detected by the fluorescence detection system probe, and probe sending the information about the amplification to the computer, computer having some softwares, those are analyzing that the sample is positive or negative, genes are upregulating or downregulating. So we are detecting the samples is positive or negative, or also know about the expression of different genes. So both types of studies we can do it here. So now we discuss about the different chemistry we are using for the real-time PCR. First one is SYBR Green and second one is the TaqMan Pro PCR. In SYBR Green chemistry, it's a fluorophore. So fluorophore only can bind to the double stranded of the DNA. So during denaturation, the double stranded DNA melted out. So there is a single strand. So no fluorophore can bind there. So there is no fluorescence. When primers polymerize, when primer anneal with the template and start amplification, so when they synthesizing the new strand, then they are creating the double stranded of the DNA. So that time only SYBR Green can bind and give the fluorescence. So at every cycles, because copy number of DNA is increasing, so fluorescence will increase with in each and every cycles. Second chemistry is the TaqMan probe, real-time PCR. Here we are using the template specific probe. So when primer anneal with the template and start amplification, then due to the 5-3' exonuclease activity, primer clip the reporter from the probe. When reporter maintains the specific distance from the quencher, then it starts glowing. It means if there is amplification, then only reporter clips out from the probe. If there is no amplification, it means there is no 5-3' exonuclease activity. That time reporter not comes out from the probe and not releasing the fluorescence. So these are the chemistry of the real-time PCR we are using for the amplification. Now we discuss about the details of the SYBR Green qPCR. So first we know about the ingredients. What types of ingredients we are using for the SYBR Green. So first we are using the SYBR Green ROX qPCR master mix 2x concentration. So that's why we are taking half of the total volume of the reaction mix. So its volume will be 10 microliter. Then primer we are using in 10 picomole per microliter concentration and its volume will be 0.25 microliter. Another primer, reverse primer, we are using in 0.25 microliter volume and same concentration. Along with we are using HPLC nucleus free water 7.25 microliter volume and template 2 microliter and total volume will be 20 microliter. And now we know about the profile of the SYBR Green PCR. So to start to activate the enzyme, because in the SYBR Green qPCR master mix, enzymes are attached with the antibodies. So to activate the enzymes and to release the enzymes from the antibodies, we incubating the master mix at 50 degrees Celsius for 2 minutes. That activates the enzyme. Then we enter into the initial hold at 94 degrees Celsius for 5 minutes. Then we enter into the denaturation main cycle of the real-time PCR. Here we go for 94 degrees Celsius for 30 seconds. Then we go for 61 degrees Celsius for 30 seconds. Then we go for extension at 72 degrees Celsius for 30 seconds. And the main cycles of the real-time PCR, we repeating this cycle for 39 cycles. Then we go for melt curve analysis by raising the temperature from the 65 to 90 degrees Celsius for 20 minutes with continuous imaging by CCD camera, and then we go for hold. Melt curve analysis needed because if there is a contamination, if is a non-specific product inside the SYBR Green qPCR, then we can easily detect it. Is there any contamination? Is there any non-specific products are there or not? This is melt curve chart. Here we can see that the y-axis showing the fluorescence comes from the real-time PCR, and the x-axis showing the temperature. At melting temperature, the fluorescence comes down if there is no contamination. All the samples fluorescence comes down at single temperature. If there is a contamination, then we can get difference in the melt curve chart. In melt peak chart, we can see that at melting temperature we get a peak. If there is no contamination, we will get single peak. If there is a contamination or non-specific product, then we can get another peak, and this peak comes out at the temperature of melting temperature. And third is the amplification chart. Amplification chart showing that at what cycle the amplification comes out from the threshold. So you can see that some are coming in 12-13 cycles, some are coming after 15. So it depends on the copy numbers present inside the sample. If copy number is high, it comes earlier. If copy number is less, it comes later. When fluorescence comes out from the threshold, that is called, the number of cycle is called CQ value or CT value. So CQ value and CT value is the number of cycles where fluorescence comes out from the threshold and machine is able to detect the fluorescence. So that is called CQ value or CT value. Now we discuss about the amplification in real-time PCR. In initial phase, because fluorescence is very less, so when it comes out from the threshold, then machine can only detect the fluorescence. Then it enters into the exponential phase, then it enters into the linear phase, and then it enters into the plateau phase. We can put more than one reaction at single time. If there is more than one product, then you will get two melting curve, because both of the product having different melting temperature. We both get both of the melting curve will be different. Here we can see that in melt peak analysis, we can see that if there is a two product, then we can get two different peak, and because their melting temperature should be different, that's why we are getting two different product there. Here we can see that if there is the two different products are there, or if there is a contamination, then we get two different melt curve there. It depends on the GC content of the product. Here we can see that there is a dimer. If there is a dimer, primer dimer inside the product, then we can get a small peak before the specific peak, melt peak of the product. So that is called, a small peak is called primer dimer. Due to primer dimer, we can get a small peak there. Here we can see that if there is a primer dimer, it can come much earlier than a specific product. But if there is, and in a specific product, we can get all the melt peak at same temperature. Here we can see that there are two types of DNA we are using for the amplification. One template is normal and second template is mutated. If GC content is high in mutated DNA, so then we can get slight shifting of the melt curve in mutated DNA case. Here we can see that we are analyzing the mutant, heterozygotes and wild type of the template for the real time PCR. Then in wild type, because wild type having high GC content and mutant type having lesser content of GC content, so that's why we are getting the melt curve earlier in mutant and later in wild type. Now we discuss about the TaqMan probe real time PCR. So first of all we discuss about the ingredients. So we are using Maxima probe qPCR master mix, because its concentration is 2x, that's why we are using 10 microliter because total volume is 20 microliter. Then we are using primer, forward primer and reverse primer in 10 picomole per microliter concentration, and volume of each primers will be 0.25 microliter. We are also using the specific probe 0.25 microliter, nucleus free water 7.25 microliter and template 2 microliter. Total volume of the reaction will be 20 microliter. Here we go for initial incubation to activate the enzyme. Similarly as SYBR Green real time PCR, then we interest into the initial hold, then we interest into the main cycle, and then we go for denaturation at 94 degrees Celsius for 20 seconds. Annealing and extension, we are using same temperature for annealing and extension at 61 degrees Celsius for 30 seconds, and we are repeating these cycles for 40 cycles. And we are collecting the fluorescence. We are capturing the fluorescence by CCD camera of the real time thermocycler. So this is the amplification cycle. So we can see that some samples come earlier, start giving fluorescence earlier than the other samples. This is showing that if we are going for the positive for deciding the positive and negative samples, then then fluorescence comes earlier in the samples. We can say that the samples having high copy number. The amplification comes later, fluorescence comes later cycle, then we can say that the samples having low copy number. If there is no amplification, no fluorescence, we can say that there is no specific template there. So now we discuss about the different applications of real-time PCR. First is gene expression analysis. So we can go for using real-time PCR, we can go for gene expression analysis. So we can check the up-regulation and down-regulation of different genes. Then we go for cancer research. In cancer research, different types of host genes are responsible for cancers. So we can check their up regulation and down regulation and their expression using real-time PCR. Third is drug research. In drug research, we can apply the drugs on any diseases, and then we can check the expression level of the genes inside the host. Then disease diagnosis and management. We can know about the copy number of the load of the viruses, load of the bacteria, load of the microbes inside the host, and we can by this way we can diagnose the sample is positive or negative. Viral quantification. Using this real-time PCR, we can quantify the viruses also. In food testing, if there is a contamination of any microbes, we can know about those microbes using the real-time PCR. GMO food, we can also go for analysis in GMO food. We can also go for animal and plant breeding using the real-time PCR, and we also know the gene copy number available inside the sample using the real-time PCR. So thank you everyone, thank you.